So this is a long chapter, so I wanted to try to go over this, not take too long doing it. So to get started, let me just, where am I? Keep having to switch between Mac and PC. Okay, here we go. All right, so chemistry of living things. And next week's labs are going to be looking at our macromolecules, just so you know. All right, now, to begin, I just started off going over these definitions. Matter, elements, compound, energy, right? So matter, anything that has mass and takes up space. This is where we're thinking of our elements. Everything's made up of these elements, this matter here. Elements, they can't be broken down into a simpler form. So you think about the periodic table. Compound, when you've got two or more of those elements put together, and they're usually in a fixed ratio. And energy is simply the power to do work, and we'll talk a little bit more about that. Now, the point that I was emphasizing by the images here was that when you make a compound, you can take elements that have very different properties when they're on their own, so sodium and chlorine, and then when they combine, they have completely new properties, all right? And that's simply by the process of bonding and how those elements are put together, all right? So the purpose here was just to remind you about some of the, tell you about some of the main elements that we'll be talking about human biology. The main ones, and you probably know now if you came to class, and I'll go over it again, of course, but the big four are oxygen, carbon, hydrogen, and nitrogen. Now, with that, so those are our main ones. You can see that's over, most of it, so 96.3%. And then there's some more coming up from these other ones: calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium. So they are, and even though we don't have a lot, so less than 4% of those, the thing here is that that little bit makes a huge difference, as you'll see as we go through the course. And then, of course, we have the trace ones at the bottom. So these are very tiny amounts, but if we don't have them, we will be sick. We will miss them. So even though their amounts are so less than 0.01% of body mass, very important to function and very often to enzyme function. Okay, now, so then going over this idea here, parts of the atom. So the nucleus is the middle, the nucleus, and there we've got protons and neutrons, and protons have a positive charge and they've got a certain amount of mass, and the neutrons have no charge, but they also have mass. So pretty much when we're dealing with the mass of the atom, it comes from the protons and the neutrons here in the middle. Now the electrons, which are negatively charged, they exist outside of that nucleus and they're spinning around here. I know it's not in 3 dimensions, but you can imagine it coming out of the screen and into the screen in every kind of direction here, but they're going around very, very quickly. Now they have very little, very non-discernible or no discernible mass. But of course their charge is negative, and that negative charge is really important to making bonds, all right? Sometimes you'll see these electrons drawn in orbitals, and those orbitals is where they are most of the time. I can't remember, it's like 90-something% of the time they exist in these orbitals, all right? Now, periodic table of the elements. Now, the only reason I bring this up is twofold, and since we've already gone through class, I'll just bring it up right away. So first of all, when you look at the periodic table, something that should strike you is that as you go through the different rows, you're getting more electrons as you move towards the right, okay? So, and we're showing them here. What we're looking at are the orbitals where these electrons reside. So you're going to get more electrons in a given orbital as you go towards the right, and you have more electrons in general as you go from top to bottom, all right? Now, what's happening is as you go towards the right, these orbitals are filling. So the first level, there's 2 electrons, and then the second and third, there's going to be 8 electrons. And then if you take physics, you're going to learn that the d orbitals are a little bit different, but usually we stop here. They work very similarly, all right? But when we're talking about what we do here, we're focusing mostly on these small atoms here and their chemistry. Now here in the nucleus, remember you have your protons and your neutrons, and the number of protons is the same for any element. It's always going to be the same, and that's the atomic number. Okay, so here you have 1, and you have 2, here 3, 4, 5, and you see that's how we number them going up by the number of protons. Now, the atomic mass, since only the protons and the neutrons actually have a discernible mass, the atomic mass is pretty much equal to the number of protons and the number of neutrons. So if on the midterm or in the homework I give you the atomic number or the atomic mass and I tell you how many protons, you can figure out how many neutrons one has, okay? All right, so yes. Now, what can vary, all right, so the number of protons doesn't tend to vary, but what can vary, not with every element, but with certain ones, are the number of neutrons. And when you have an element with different numbers of neutrons, we call those other atomic masses the isotopes, okay? So here you can see helium. And here you have 1 proton and 1 electron, no neutrons. But deuterium has a neutron, and then tritium has 2 neutrons. This one is the one that's radioactive. So radioactive ones are isotopes that aren't stable, so they will decay. How that works is something that you will learn in physics if you take physics. But for now, just understand that some of those isotopes are not stable and they decay, so they kind of lose their neutrons as you go. So here, tritium will decay to deuterium, okay? The other one that's very common here is carbon. So our main one is carbon-12, but there's 2 isotopes, carbon-13 and carbon-14, and 14 is the one that's hot. We use it in the lab. Not as frequently as we used to, but certainly when I started out in the lab, using radioisotopes was very common. Now we have other ways of doing things. The technology has advanced. We don't need to use these things as frequently. Carbon-14 was never one that worried me too much. It's not too dangerous, but P32, so the radioisotope of phosphorus, and sulfur, I-125, those ones are pretty scary. But especially the 125. Anyhow, I digress. So radioisotopes are the ones that are radioactive, and those are the ones that you worry about if they're really radioactive, like what I was just discussing. But carbon-14 is pretty common in the environment, and because of that, it can be used for dating fossils. So if you go on and you take anthropology or other courses, you'll learn a bit more about that. But it decays at a certain rate, and that's how you can use it to date fossils. Now, in human biology, we use radioisotopes in diagnostic imaging and cancer treatment, power supplies for pacemakers, and that kind of thing. So they have a real-world application in regular old human biology, okay? Which is why we mention them here. Okay, now, so when I was talking about those orbitals, you think back on those orbitals I was just discussing on that little piece of the periodic table. Now, those electrons that are in those orbitals are ones that drive a lot of the chemistry of these elements. And they're particularly the ones that are found in the outermost orbital. So if we go back here, right? I said I wasn't going to do this, but here I am. So the ones in the outermost orbital, these are the ones that are going to be driving a lot of the chemistry, because it works out that these elements would like that outermost orbital to be full. All right, so here it would like to have 8, okay, or here it would like to have 2, so electrons. So it drives some of the chemistry, and we have different types of bonds depending on how those electrons are used and what happens with the electrons that are spinning around the atom. So I'm going to go over again the 4 main types of bonds that we consider: covalent, ionic, hydrogen, and van der Waals. So with this, and I'm going to just move forward in my notes here to make sure I'm telling you everything that I'm supposed to. Yeah, there we go. So covalent bonds, this is where 2 atoms will share the electrons equally. Or I should just say, right now I'll say, share the electrons, all right? So we've got the 2 flavors, polar and nonpolar, all right? So in covalent bonds, the electrons are shared. These are very strong bonds, and they trap a lot of energy. So by this point, you should know that we store energy in bonds, right? Now, so when I talk about these covalent bonds, the first one's the nonpolar bonds. these are the ones where the electrons are shared equally, and it tends to be when a molecule is made up of one or more of the same atom. So hydrogen, it has two hydrogens. As you know from the periodic table back here, hydrogen, right, and helium, right, will share these electrons until we have two electrons in the outermost orbital, right? Now, here we go back here. I'm thinking about something else here, so I'm a little distracted. So here they're sharing these equally. But now, if these are not the same atoms, right? So if we're talking about two different types of atoms, they may not share those electrons equally, but they will share them. So, and that's where you get polar covalent bonds. And in this case, the electrons are shared unequally. So, and the reason that is, and we'll go back to this periodic table, if you see something like we had oxygen and two hydrogens there, it's water, right? So look at oxygen and look at hydrogen, right? So you've got one electron here. Here you've got, let's count them, 1, 2, 3, 4, 5, 6, 7, 8. We have 8 electrons. So if you think about this, this thing is, first of all, a bigger molecule, right? Or a bigger atom, I should say, because it already has more electrons. And it's also more negative, right? Because each of those electrons has a negative charge to it. So this one has essentially, it's much more negative because it just has more negative parts to it. And this one, even though this electron is still negative, overall this atom is not as negative, right? So if you look at oxygen, it would like to have 2 more electrons here. And hydrogen just wants 1 more electron. So what ends up happening is that hydrogen in water shares those electrons, but they shares them unequally. They're a little bit more pulled toward the oxygen simply because it's a bigger molecule and it's pulling those electrons more strongly. So what ends up happening is that with the water molecule, the hydrogens are giving their electrons a little bit more to the oxygen, and the oxygen pulls a bit more on its electrons versus the hydrogen. And what ends up happening is that one end of the molecule is a little bit more positive, whereas the other one is a little bit more negative. So it's not really charged. It's not a charge. It's not like stripping electrons like the ionic bonds that we're about to review again. But this side is, the charge distribution or the distribution of electrons across this molecule is not equal. So here, this one, we can say it's a slightly negative side. This is a slightly positive side. So it's almost like, and I like to use, it's not a magnet, but you could think about it as similar to a magnet, because a magnet has a positive side and a negative side, the 2 poles. And this is why we call these polar bonds, and these give rise to these molecules that are polar molecules, okay? So they contain polar covalent bonds, which is unequal sharing of electrons, okay? So even though they are electrically neutral, the charge distribution with those electron clouds is not equal over the molecule, all right? So this is going to come back when we talk about our other bonds, all right, later on. So keep that in mind. All right. Now, the next one are the ionic bonds, and this is where the electrons are not shared, right? So one atom is so negative that it actually is so close to being full in its charge distribution, or its valence electrons, it will actually pull the electrons off of the other one, right? So here we have sodium and chloride. So sodium only has 1 valence electron, and chlorine has 7. So it would really like to have that last one. And if sodium loses that 1 electron, well, it loses its electron, but its outermost orbitals are now full. So it's actually kind of happy, right? So these bonds, they're weak. As you know, if you put salt in water, it dissolves. So sodium and chloride come apart. So they're weak bonds. But they are an attractive force between a positive and a negative charge. So here, the positively charged ion, right, forms when an atom loses its electrons, and the negatively charged ion is when it pulls the atom who pulls the electrons towards it. So in the end, notice now the valence shells are full. We have a positive charge and a negative charge, but these molecules are very stable like this, and they make a bond between each other. It's called an ionic bond. Okay. So remember, sodium is the cation, right? It's positive, so positive cation, negative, the chlorine here, that's the anion, right? Okay. Now, hydrogen bonds. Now, this goes back to our polar molecules. So polar molecules, especially these ones here, like water, right? They make these things called hydrogen bonds. These again are weak bonds, but they're an attraction between polar molecules, all right? So here we have water, and we have a sort of like positive side on one side, and here we have ammonia. And ammonia is similar to what's going on with water, right? That nitrogen is, you know, bigger, has more electrons, and it pulls those electrons a little bit more towards itself from the hydrogen, and it also is polar. So these hydrogen bonds are formed between polar molecules like this, and it's very common for these to be, and that usually the hydrogen is in the middle, so between oxygen and hydrogen, nitrogen and hydrogen, sulfur and hydrogen, those are the main ways you're going to see hydrogen bonds, all right? All right, flip through here, make sure I'm not missing anything that we discussed. Okay, now, the other one, which is important in biology, weird to understand, but it has to do, think about those polar molecules, right? So how you can visualize how in a small molecule like that electrons are not distributed equally over the molecule. Well, this can happen with a larger molecule as well, and it creates hotspots of positive or negative charge that can interact with hotspots of positive and or negative on another molecule. And these interactions are called van der Waals. It does not have to be a hydrogen bond, but it's sort of, you know, the changes in the way those electrons are flowing around the atoms of a molecule will create these areas. And a practical purpose or practical use, I mean, other than things that you can't see, but something you can see, van der Waals interactions in action would be how a gecko climbs up a wall, which is why I have this little guy here, okay? All right, so now the thing is that, as you'll see, all of these interactions take place in the shape of a molecule. So this is a protein, and we've already gone proteins in lecture, so you can already get a sense of, you know, how all of these types of bonds are at play in a protein structure. And they're also all at play in the structure of DNA, and they're all at play in the structure of the other molecules that we have discussed, all right? So what happens is that molecules have a certain shape, and they interact with each other because of that shape that's induced by the different bonds and arrangements of the elements in a molecule. And I mentioned this one here. We'll talk later on in the course about how we have receptors for some proteins, and they allow for the cell to carry out different types of interactions inside. And here I'm showing you, and they can be mimicked by drugs, all right, so in human biology, right? So we have natural endorphins and morphine, and they both bind to the same kinds of receptors, and they have similar parts to them that allow for the drug morphine to give a similar effect as a natural endorphin might, okay? And in fact, a stronger effect. It's much more efficient in the way it binds, okay? Now, moving on, all right, so water, right? So I had mentioned to you the four critical molecules in the body are carbohydrates, lipids, proteins, nucleic acids. And we're going to talk about them again in a few minutes. But water would be the fifth. Not that it's a biological molecule, right? But it's important to biology, right? Because we are, as you can see here, 60% water, and it drives the chemistry that goes on in our bodies. And you can think now that I've gone over the chapter, right? We have hydrophilic types of macromolecules, hydrophobic types of macromolecules. You're 60% water. We have ionic bonds that can dissolve in water. We have ions then in the body. So this is why water is so important, because it drives all of these things that are happening. And water itself, hydrogen bonds, all right? You can see that it has the oxygen and the hydrogen, and it can hydrogen bond to itself. And because of that, it has a lot of interesting chemistry. Okay, so yes, now I'm going to go through the properties of water. So the first property of water is sort of the way it behaves, its chemical nature at different temperatures. So first of all, it expands on freezing, and that's because of the lattice that's formed with these hydrogen bonds. So it's actually less dense when it's frozen, floats, right? And very importantly, at our body temperature, it's a liquid. Okay, that's really important. And for example, we are overall 60% water, but our blood is 90% water. So you think about the transport of molecules throughout that blood and just say they're not dissolving in water very well, which we'll talk about later. So if they're a hydrophobic molecule, like those steroids that we talked about today in class, all right? And I'll go over it again. Okay, so water is in the intracellular spaces and in the extracellular spaces, so it's really driving the chemistry. Now, so this one isn't as important to us unless we're looking for food and we're fishing in the winter, ice fishing. But for a polar bear, the fact that ice floats is critical because its main food source lives in the water, and this is how it hunts. And now with the loss of ice, polar bears are really suffering because they can't get out onto the ice to get their main food source, which is the seal, all right? And they spend most of their time on the ice, actually, the polar bear. Now, the second property of waters is that it moderates temperature. It's a really good heat sink. All right, so which is a really good thing for us because we're mostly water, and it helps us to maintain our homeostasis, if you remember that term that I used already in chapter 1. So it helps prevent rapid changes in body temperature. And in the real world, outside of human biology, this is why it's always a little bit cooler along the coast in the summer because it takes a little longer for the water to heat up. And in the winter, it's a little bit warmer because it still has the heat left over from the summer. So it's a really moderating effect along the coast. And it happens similarly in our body. Now, the other thing is since it's such a really good, it's really hard, it takes a lot of energy to change the temperature of water, it's also very good for cooling. So if you go back here, whoops, and you see water, right? To break these nice hydrogen bonds and change liquid water to vapor water or water vapor, it takes a lot of energy. And this is why we sweat. So if we're hot, we sweat, that liquid sits on our skin, and as the water evaporates, it's going to pull the energy out of our skin and cool us off. So that's another really important thing, and it helps us moderate our temperature as well. So when we get really hot, right, we do that. All right. The other thing is water is cohesive. We don't really think of it too much when we're talking about human biology, but it's really important for things like the water spider, right? So how it walks on water. And cohesive just means that water sticks to itself because of those hydrogen bonds. Now, it's also really important for plants because they don't have a heart to pump water or fluids around. So they rely on the evaporative properties of water and the cohesiveness of water to pull water from the roots where they essentially drink or they take in water and get that water up to the leaves because those leaves also need the water. So what ends up happening is that there's these columns of water inside the tree. And when the top molecule evaporates, it pulls the next one up, and then it evaporates, and it pulls the next one up, and so all the way down to the roots. And sometimes that column of water will snap, and you can hear it. Not without a stethoscope, but you can hear it if you would put a stethoscope to a tree. You would be able to hear the snapping of this. All right. Okay. The other thing, like we already discussed, water is a really good solvent. I didn't use the word solvent, but I said that the salts in our body dissolve because those ionic bonds are weak. So water is a really good solvent. So solvent, what is it? It is a liquid. We mostly think of it as a liquid or a medium which dissolves other substances. And the solute is the thing that's dissolving. So here the solute is the salt, sodium chloride, and the solvent is water. So why is it such a good solvent? Well, remember, it's polar. So it has a positive, slightly positive end and a slightly negative end. So when it's dissolving salts, the slightly positive ends will sort of bore into this and surround the chloride, for example, and the slightly negative sides will surround the sodium. In this case, right? So it's positive charge. So this is how it works, right? And this is why it's so easy for water to dissolve salt. Now, this brings up this idea of hydrophilic and hydrophobic, right? So like dissolves like, right? So hydrophilic, that's water loving, right? So these are things that are going to dissolve well in water. So these will be polar because water is polar. And ionic molecules dissolve very well in water as well, because they are also polar, not quite like what we were discussing, but since they are ions, they, without even thinking about it, they have poles, right? Positive and negative. Now, hydrophobic, right? Those things are water-fearing. They don't dissolve very well in water. So non-polar molecules or neutral molecules will also not dissolve as well in water if they don't have a sidedness like a polar molecule would, okay? So those things are hydrophobic. Alrighty. Now, large molecules can also dissolve in water because they do have areas of charge, positive sides and negative sides, or hotspots of these charges, and water molecules will arrange themselves around large molecules, such as proteins, which we already discussed in class, and it will allow proteins to dissolve. Okay. Now, the last thing is that water is participating in our chemical reactions, and I went over this so many times in lecture. So synthesis and the breakdown of molecules in the body requires energy in water, or involves energy in water, all right? So remember, the bonds store energy. So when we are building a molecule, so we're going through anabolism, right? So remember, metabolism involves anabolism and catabolism. And we are building those bonds. We're going to store energy, and we're going to release water, okay? Now, on the other end, when we're breaking molecules apart, we're going to release that energy. We need to use the water, and then we get our molecules or our atoms apart. So this part where we're splitting the water and we're using it to break these molecules is called hydrolysis. And when we're building molecules, we call it a dehydration synthesis, okay? All right. Now, in this case, we're talking about a carbohydrate, which we're going to talk about in a sec, all right? All right, now, to reiterate, why do we talk about pH? And that's because the chemistry that goes on in the body is very dependent on pH, as well as particularly protein folding. And hopefully by now, after being in lecture, and I know I'm reviewing it, but you know that the shape of proteins is really dependent on the pH. So what is pH? Well, let's get our terms here. So an acid is any molecule that can donate hydrogen and increases the hydrogen ion concentration in the solution, whereas a base is any molecule that can accept hydrogen and decreases the hydrogen ion in a solution, okay? So the pH scale is simply an easy way to express the concentration of hydrogen. And the reason we do this as scientists or as biologists is because water can dissociate, right? It forms a strong base and a strong acid, right? So we have hydroxide, which is the strong base, and hydrogen or protons, which would be the strong acid. And I apologize, my phone is ringing. Just make sure that.
Call from Toronto OM.
All right, sorry, turned off my phone. Okay, there we go. Now, wait, where was I? Oh, yes, but this does not happen very often, all right? It's a very rare event, but it does happen. But it's enough for scientists to be motivated to create this pH scale. So this pH scale, it is the negative log of the hydrogen ion concentration, okay? And we're dealing with concentrations from 10 all the way up to, so basically 10 molar, all the way to negative 14 molar, all right? And it's the negative log, so this goes from 0, because the exponent here is 0, it's a negative log of the hydrogen ion concentration, all the way to 14. Now, if you look at it, you can see that the lower the pH number, the more hydrogen you have. Okay, and that's simply because it's this negative log, so it's like counterintuitive. But remember, the lower the pH, the more hydrogen. Okay, neutral down the middle is pH 7, and simply it's neutral because look at the concentration of hydroxide and hydrogen here; it's equal, so therefore it's neutral, right? So the same thing here: one molecule breaks into one of these and one of these. So at this point, it is neutral, so that's pH 7. Now, anything less than 7, it could be 6.9999999, it will still be more acidic, all right? And anything above 7 is going to be basic, all right? So even if it's 7.000000001, it is basic because it is higher than pH 7, okay? All right, yes. Now, the other thing, where do we encounter this? With different pHs in the body. Overall, we think of the human body as being around neutral, all right? But that doesn't mean that everything is pH 7, all right? So it's not really quite like that. I mean, probably more like 7.4. But even so, we've got variety, right? So the small intestine is a little bit basic of pH 7. Blood is a little bit acidic of pH 7, but a little bit more acid than 7, okay? So around 7.4, which is why we think of our pH being around 7.4 because of blood. But you can see urine and saliva are more on the acidic side, right? And then here in gastric juice, we're all the way down at 2, and sometimes it's quoted as 1, right? So depending on your source, but it's very acidic. Now, and I had highlighted in class that, well, think about where does this gastric juice go that is at pH 2? Well, it goes into the small intestine, which is buffered, well, buffered, I already used the word, but it goes up to pH 8, all right? So the body is in tune with that, and changes have to happen fairly quickly, again, because of proteins, because we have proteins in the stomach, we have proteins in the small intestine. Okay. Now, I really want to hopefully make this have sense to you, but it is a log scale, this pH. So that means every unit of pH is 10 times different from its neighboring pH, okay? So if we use pH 7 neutral as 1, if we go to urine, all right, which is pH 6, it's 10 times more acidic, not just 1 times more acidic, 10 times more acidic, okay? So every time we go at 1 pH unit, we are going a scale of 10. So if I were to ask you, what is the difference in a pH of 5 versus a pH of 2? Well, let's do this. So we're starting at 5. So 10 times, 100 times, 1,000 times. So that means that at 2, we have 1,000 times more hydrogen than we did at pH 5. Okay, so hopefully that makes sense to you. So you're going by tens, right? Now, remember, the lower the pH, the more hydrogen, all right, the more acidic. The higher the pH, the less hydrogen, the more basic, all right? Okay. Now, how do we take care of those rapid changes in pH or any changes in pH, for example, in the body? We have buffers. And buffers, basically what they do is they minimize the pH change by keeping things a little bit more stable. They're hiding the excess hydrogen or hiding the excess OH, all right? And that's what their job is, okay? So in the human body, we have one critical buffer that is, we consider the biological buffer, and that's the carbonic acid bicarbonate buffer. So this is bicarbonate, it's carbonic acid. And this is a reversible reaction, all right? So say blood, it's buffering at around 7.4. If we get too much acid, that will combine with the bicarb and make carbonic acid. If it's too alkaline or too basic, the carbonic acid is going to separate and we're going to release some of these hydrogens. So that's going to buffer it out. Now, if you want to see a little review video, I encourage you to watch that. It works, okay? It tells you everything you need to know, again, all right? Now, energy. When we talk about energy, we talk about the energy of our bonds and the energy when we release break our bonds. So energy, again, is the capacity to do work. Potential energy is stored energy, or the energy of position, if you take physics. And kinetic energy is the energy of motion, or the energy that's released when we break bonds in biology. Okay, so potential energy is stored in bonds, kinetic energy is released. All right, and we used Michael Phelps here as the example. All right, so he's got a lot of energy stored up in his muscles and all the molecules there, and when he gets going, he's going to release that energy, and that's going to power his swim. All right, so that potential energy can be changed into kinetic. This example here is trying to take that idea of energy and show you how we do work and how we need energy to do work, okay? So the old adage of if you can't start your car, put in some energy, push it up a hill, and then as it goes down, that potential energy at the top will be converted to kinetic energy and help you start your car, all right? The same thing happens with the electrons around an atom. The further out from the nucleus those electrons are, the further towards the nucleus they could fall, and that would release more energy, all right? Now, in the body, what do we think about? Well, we have chemical reactions. We're talking largely right now about dehydration synthesis and hydrolysis. So these are facilitated in the body by things called catalysts, and those catalysts are proteins called enzymes. Now, why do we need them? Okay, well, you know, to get a reaction going, you need to put a certain amount of energy in, and we're going to talk about that in the next chapter before the midterm, all right, so Monday. But basically, we're putting in energy, and that's from the food we eat, all right? So we need to put a little bit of energy in, okay? And you'll see that when we go over this, we're going to do it in ATP. That's our energy currency. That's no secret to you, but we'll show you how we do that. So a little bit of energy needs to go in. And when you're looking at it in terms of chemistry, we say that's the activation energy, all right? Now, in order to make a reaction a little easier, we can use a catalyst. What a catalyst does is it helps to make a reaction happen, and in so doing, it lowers the amount of energy required to do that. And the human body does this all the time. Our catalysts are proteins, okay? So here we have an example of a reaction that may be taking place in the body. So here we're linking two molecules. We have the enzyme, the biological catalyst, will facilitate this because it will help the molecules get in the right orientation and then bring them together, all right? Bring them together, help that reaction take place, and much more efficiently than if we let these molecules do it randomly on their own, okay? And then the key thing about a catalyst, such as these enzymes and any catalyst, is that it's not going to be used up in the reaction. And at the end, the catalyst is the same as it was when it started, right? So meanwhile, while things might be happening to it during the reaction, at the end of the day, you're going to release it and it's not going to be changed overall by what has just happened, okay? So in the body, every reaction that we think about, most of it, I mean, it's hard, I'd be hard pressed to find one that didn't apply to this, but it's really, you know, these all require enzymes. Okay, surely things do happen in the body without them, but for the most part, we need our enzymes. Okay, now let's talk about these biological molecules, and of which enzymes, being proteins, are in there. So here we have organic molecules and inorganic molecules, and the organic molecules, those four big ones that we talk about, are based on carbon. They're pretty large and pretty complicated. Some are more complicated than others if you think about it in terms of their complexity, all right? But they're large and complex, and there's lots of covalent bonds as well as other bonds, all right? Now, why are they based on carbon? Well, first of all, there's 4 valence electrons, which gives a potential of 4 different bonds, all right? And it's somewhere in the middle. It's not going to want to give up its electrons too easily, and it's not going to rip electrons apart from something else too easily. It's nice sort of think about it as neutral, in fact, that it's not aggressive, all right? Now, the other thing is that since it can form these 4 bonds, these bonds could be single bonds, double or triple bonds, all right? And here you can just see, even between just 2 carbon molecules, you can see that, right? And very importantly, obviously, it can form covalent bonds with itself as well as many other types of atoms. Primarily, though, we think about it in terms of oxygen, nitrogen, hydrogen, and sulfur when we talk about the human body. Now, the other thing that can happen is that these molecules can vary in their length, right? They can vary in the position of double bonds. They can be branched. They can be ring structures. Can do all kinds of stuff. And finally, the size can vary incredibly. We can have little teensy tiny molecules like methane, which is 1 carbon, or it can go up into fancy structures. I mean, of course, carbon nanotubes are not natural to the human body, but it's just showing you the potential of carbon to form these huge complex molecules, all right? Now, with that, these carbons in these molecules have certain structures that make our main 4 biological molecules. So when these molecules are put together three types, the carbohydrates, proteins, and nucleic acids, are considered polymers. So what means when we have a polymer is that we have a series of repeating units, which are called monomers, that come together to make the polymer, the bigger structure. One of these is not a polymer. Those would be the lipids or the fats, all right? So we're using the Lego analogy. We have different colors, so while as these monomers are very similar in their shape and identical, they can vary a little bit in terms of their properties, and in this case, the color of the little Lego blocks, all right? So when we are forming a molecule, we're forming bonds. Remember what we're doing is we're joining bonds, we're making a bond, we're gonna lose water, we're gonna add energy, and that energy is gonna be stored up in that bond, okay? So that's our dehydration reaction, our dehydration synthesis, okay? And then conversely, when we're breaking down the polymer, we're gonna break that bond, we're gonna add water, and we're gonna release the energy, and that process is called hydrolysis, because we're splitting the hydrogen in order to break the bond. One hydrogen is going to go here and the OH is going to go on the other monomer to now separate them. Okay. And when we make the polymer, it's repeated dehydration synthesis. And when we break it down, it's repeated hydrolysis, releasing energy each time. Now, in terms of this energy, where do we get the energy? All right. Well, we get it from our food, which we'll go over shortly before the midterm, but it gets transferred to ATP. And ATP, likewise, when we release energy from ATP, okay, so remember, hopefully you remember from way back somewhere, we have 3 phosphates joined together. We split off that last phosphate, we call it the high energy phosphate, that we need water to do that, so we're going to use water, but we're going to release that energy and we're going to use it to do work in the body. Okay, and then we can recycle it. We can put a phosphate back on. We're going to store up some energy, and we're going to release water. Okay, so keep that in mind for when we talk about that on Monday. Now, our first biological molecule we talk about here are the carbohydrates. So carbs, we know we get lots of energy from that. If you're an athlete, you know you should eat your carbs, right? If you're an athlete, you can eat more carbs than the average person because there's a lot of calories in some carbs, right? But we'll talk about that later, all right? Now, here I'm showing you a sugar molecule, glucose molecule. That's because most of our carbs are based on this one sugar, which is glucose, right? Surely we eat other sugars, and we'll explain that, right? So the cool thing about carbohydrates, they are polymers, and they're polymers of single sugars or monosaccharides, okay? The main one, like I said, in the body where we store energy is glucose, this one here. But in our diet, we get these other ones, fructose, galactose, ribose, deoxyriboses, and these pentoses here. And we use these, and these are actually converted to glucose for our use, but we definitely eat these. This one we love because it's really sweet, okay? But they are active. This one, the carbohydrates are actually active already as a monosaccharide. It doesn't have to be a polymer. The other ones are active as polymers. And also as disaccharides, all right? So 2 monosaccharides put together. So glucose and glucose put together makes maltose. Glucose and fructose together makes sucrose, right? This is lots of additives. There's lots of places that use sucrose, right? And the other one that I mentioned in lecture was lactose, and lactose is a molecule of glucose plus a molecule of galactose, all right? Now, the polymers. So polysaccharides, many saccharides, many sugars put together. And we're pretty much based on glucose here. So we have starch that we use. These are made by plants. It stores energy in the plants, and we definitely get that energy out of the plant, right? Because that's the sun's energy is stored up as starch in plants, and then we use it for ourselves. And then if we take in too much of that kind of stuff, as well as animal stuff, it's glycogen, we make it as well. So we can get glycogen from animals as animal. Then we store energy because we eat, if you eat meat, if you're vegetarian, you'll eat milk. If you're vegan, obviously you're not going to be taking in the glycogen, so you're going to be getting you'll be making all of your glycogen from starch, okay? And then cellulose, right? We can't digest it. It's a structural component of plants, but I mentioned in class it's important for us to ingest it, even though we can't digest it, all right? Okay, so what are the differences in these forms? So starch, again, glucose molecules, it can be in this nice sort of helical form, which is amylose. You can have branches on that, amylopectin. Glycogen looks very much like amylopectin, just more branches, but very similar. We digest both equally easily. Now, cellulose, it's a little different, and it's also based on glucose, but it's a little bit an isomer of glucose. It's isomer meaning slightly different form. We'll look at it that way. So we use alpha glucose to make starch and glycogen, but cellulose is made out of beta glucose. So you can see it's just a little flippy-do here, this OH and the H. And it makes a big difference in the properties. So this makes these long chains. It makes fibers like this. That's why, think of celery. We've got these cellulose fibers, and we're not very good at digesting it, but we need it for our diet. This is what we call fiber. All right, it's very important for our diet. We'll talk about why later in the semester. Okay, and it's just because it's a different form of bonding that takes place with this beta glucose, and we just don't have the enzyme needed to break it down. So if you look at it at this level, so starch and glycogen, the molecule looks like this, and if it's cellulose, you'll see that the OH, the hydroxide, it sort of bounces from side to side, and we don't have the enzyme that fits in here to break this bond. So animals that use cellulose as their prime way of getting sugars, they actually have bacteria to help them digest this. And the bacteria, or the microbes, that's not always bacteria, but the microbes have the enzyme needed to break this down. It will release the sugars so that the animal then can make glycogen. Okay. And then chitin, this is one we don't think about too much because most people here don't eat insects, but there's definitely people in the world that do eat insects. It's a very cheap form of getting your carbohydrates. It's very, very, very cheap. There's a lot of talk now about how we might be eating more of these, and I'm sorry that disturbs you, but that's just the way it is as we try to feed a growing population. But here, the chitin is a structural component as well, but of arthropods, so of insects, and it makes up the exoskeleton. And this is a cicada. Those are those, the insects in the summer that you hear rattling in the summertime, shaky, shaky, shaky. That's what these guys are. You don't see them very often. They're really interesting, but that's what they are. Now, where do we encounter it most in our society? Well, they use it to make surgical thread, okay? The nice thing is that it makes these nice fibers because it's a structural molecule, but it does dissolve with time, right? So that's why if they put stitches inside of you, those stitches will, they don't need to go back in there and pull the stitches out because they will eventually dissolve, right? And it's also based on glucose, but the glucose has a little add-on here. So it's a slightly different molecule. Now, lipids. So remember, not polymers. They come in 3 flavors: fats or triglycerides, phospholipids, and steroids, all right? So fats, triglycerides, energy storage, phospholipids, cell membranes, steroids, they've got those ring structures. I think most of them, in our case, will encounter them as hormones. So the triglycerides, okay, they are not polymers. This long chain of carbons confuses students to no end, okay? But this is a molecule called a fatty acid. It's not a polymer. It's just a string of carbon molecules with an acid group on the end. Now, what happens in a triglyceride is that we've got 3 of these fatty acids that are joined to a molecule of glycerol via a dehydration synthesis. So if we then take this, we make our 3, and these 3 do not have to be the same. They can all have a different length. Typically, we run 16 to 20. This one is called palmitic acid, and they're given different names based upon the properties or the length and the type of bonding. So here, these ones are all single bonded. And here we have a triglyceride. And there's lots of energy stored up in these bonds, and actually a lot more energy than we can get from a glucose molecule, all right? Now, if all of those bonds are single, we call it a saturated fat, and that's very typical of animal fats. They form a solid at room temperature, right? Butter. Whereas an unsaturated fat, some of those fatty acid chains have some double bonding going on in them, and they will bend. And because they bend, they don't pack as nicely. So we think of these as oils, right? So they're liquidy, right? So these are very common in fish and very common in plants, okay? Now, I talked today about trans fats. I'm not going to talk too long about it here, but basically what happens is that in our foods, because this is a little cheaper, what they'll do, if they want this and they have this, it's cheaper, they'll hydrogenate these bonds, all right, to try and get more saturated fats from an unsaturated fat, because these are cheaper than having lots of cows around or whatever, wherever you're getting your saturated fats from. And that can then be used to make things that we like to eat, all right? The problem is in the process of adding hydrogen, or in the process of hydrogenation, it creates these things called trans bonds because it doesn't always work as well. And the structure of this is a little bit hard for us to break down because it's not something we naturally encounter too frequently. So we're not really well equipped to do it. We can, but just not at the level where, you know, if we have a lot of trans fats in our diet, at the level where we need to do it. And this causes problems in the body, such as heart disease, all right? We'll talk later in the semester why. So this is my little pontification here. If you're trying to lower your trans fats, look at the ingredient list. Don't look at this baby here because they sort of futz around with the serving sizes to make it look like they can say 0 trans fats. And this is because it's based on a legal percentage in which they must report at. So if they make the serving size smaller, it changes the percentage of trans fats. But if you look at the ingredient list, they have to say this: hydrogenated oils or partially hydrogenated vegetable oils. That means that there will be some trans fats there. Okay, all right. The other one, phospholipids, look very, very similar to the triglycerides, right? Over here, right? So we've got our triglycerides. So you're going to have the same thing. You're going to have your glycerol and you're going to have fatty acids, but only 2 fatty acids now. Okay, so let's go here. We've got 2 fatty acids. And then and this part is very hydrophobic, all right? Because remember, lipids, they're fats, they're not very dissolvable in water. I didn't highlight that, but I think you know that. If you try to mix oil and water, you know that they don't like to mix. But when you have a phospholipid, all right, you have a different property because this third position is occupied by phosphate. And look at there's a charge on there, right? Remember I said charged molecules are hydrophilic. All right, and here we also have another charge on this particular one. There's a nitrogen here too. But let's think of the phosphate head group. This is hydrophilic. So you have a hydrophobic part of this molecule and a hydrophilic part. So we see the hydrophilic head and the hydrophobic tails. And what these do, because like dissolves like, hydrophilic will interact with hydrophilic and hydrophobic with hydrophobic, or polar with nonpolar, polar with polar and nonpolar with nonpolar. What ends up happening is that these things, when you've got enough of them, they self-associate and they make a membrane. Okay, so the hydrophilic side will be facing that 60% water that we're made up of, and the hydrophobic side, because it doesn't like water, will associate with itself. And it does this spontaneously, no energy required, all right? And we will talk in chapter 3 about the membrane, the cell membrane, the plasma membrane in more detail, okay? But it's largely formed by phospholipids. Now, the third lipid is the steroid. Doesn't look a bit like the other 2, but it's still a lipid. It's hydrophobic, doesn't like water. It's based on this ring structure. You've got 3 6-membered rings and 1 5-membered ring. They all have this in them. Okay, what differs between the different steroids are these side groups, which are called functional groups. The reason they're called functional groups is because those are the things that differ between these steroids and grant them their function, all right? So look at estrogen and testosterone, all right? Not that I want you to memorize the shape, but you understand that they have that steroid group, and those functional groups cause them to behave very differently in the body. And we'll talk more about these, particularly the hormones, when we do endocrine later in the semester. Now, the third one, the proteins. The proteins, these are polymers, and they are made up of monomer units called amino acids. The amino acids are the same except in all components except for one, all right? So we have in an amino acid, we have an amino end and a carboxyl end, and these are joined to the alpha carbon or the main carbon, and there's a hydrogen attached to that, but we have the amino and the carboxyl. And this is the same for every amino acid, of which we have 20 that we use in the body, all right? There's others in nature, but not ones that we use in the body. The fourth position on this carbon, remember it likes to form 4 bonds, is the R group. And this R group will differ between these 20 amino acids. Now, when we form our protein, these amino acids will join to each other with, again, a dehydration synthesis. We're storing energy here. We're in dehydration synthesis. We're losing water. And we will join these things together. And you'll notice we have an NCC, NCC, NCC. This is considered the backbone of the protein. And here, the side chains, or the R groups, these are going to give different properties to the protein, all right? So how these are arranged, and it helps the protein fold, all right? This bond is a very special name. It has a special name. It's a special bond because it has some particular properties I don't really talk about here, but I want to tell you that it's called a peptide bond, right? Peptide bond. It joins the amino acids, the monomers of the polymer, which is the protein, okay? Now these R groups, they're very different on these 20 amino acids, and all I want you to remember about that is that some of these are nonpolar, so since they're nonpolar, that means they're hydrophobic, right? So these tend to bury themselves with other hydrophobic ones, and very often they're on the inside of a protein. The other ones, they can be polar, so they like water, or they're charged, okay? So these can be on the outside of a protein that's existing in water, all right? And these will drive the folding of the protein. If you remember from class, we have 4 levels of folding, all right? So here I was talking about, look at these 2 proteins, this string here, this is the backbone. You can see that there's these interesting shapes that these proteins take. It's very critical, that shape. That's why, again, pH is so important, because it's dependent on the pH. And these are so critical because it allows these proteins to interact with each other, like this antibody interacting with the flu virus, okay? Now, I went over the functions, the main functions that we consider of proteins, and of course, enzymes are the first one that we mentioned. I already mentioned in the passing, all right? I also brought up structural support, such as collagen. Transport proteins we find in blood. We use them to cross membranes. Storage proteins, such as casein and milk, communication, hormones, neurotransmitters. The 6th one, immunity, such as antibodies, and movement, like we have proteins, and we'll go over all of these in detail when we do muscle. Now, the shape of these is critical for this, all right? And it's influenced highly by pH, as well as other things that we'll talk about. So when we make these complicated, beautiful structures, I think they're stunning. I know I'm biased because I'm a biochemist, but the first level of structure, all that is the sequence of those amino acids. That's all it is. There's no fancy shape here at all. And it's stabilized by those peptide bonds. Now, the second level, the secondary structure, this is when this chain starts interacting and folding on itself. And it makes some little shapes, sometimes helices, which is called alpha helix, or like sheets called beta sheets, beta pleated sheets. And it's stabilized by interactions not between the R groups, but between the backbone molecules, right? So remember, we had oxygens, we had hydrogen, we had nitrogen that's available. And if you think back on your hydrogen bonding, those are some of the key atoms that are involved in hydrogen bonds. And this is what's going to stabilize these secondary structural features, okay? So those are interactions between the backbone atoms. All right, now tertiary structure, now this is where those R groups become important, because they are going to create the overall folding of the peptide, all right, or the protein, all right, peptide a little smaller, or if when we have tertiary structure, we call it peptide, all right. So what's happening here is the overall folding. You can see here in this tertiary structure how the secondary folding, these helices and these sheets, come together through the tertiary interactions, and those are between the R groups. Now, what kind of bonds can we have? Well, every type of bond that we talked about. We can have hydrogen bonds, we can have covalent bonds, which are called disulfides, we can have ionic interactions, we can have interactions between hydrophobic regions or different hydrophobic types of R groups, all right? So every type of bond that we discussed would be at play here in the tertiary part. Now, every protein has primary, secondary, and tertiary structure, and some proteins are active already at tertiary, right? Now, some of them need to be joined with other proteins to make a functional machine or a functional unit, and that's when we get quaternary structure, when we get peptides coming together, and they will now work together as a functional unit. So here, this one's threatine. This one is not functional until 4 different peptides come together. And how are they stabilized? Well, they're stabilized by the R groups. So their interactions are identical to what we just talked about for the tertiary structure. So every type of bond is up for grabs here, all right? And which ones are at play will depend on the sequence of those amino acids and the folding of the peptides, all right? Polypeptides. So again, primary structure is the sequence of the amino acids. Secondary structure is local folding, alpha helices and beta sheets. Tertiary structure is the overall folding, okay, the 3-dimensional shape. And quaternary is when we bring them together. Now again, pH is so important because the charges and the properties of those side chains will be affected, as well as the hydrogen bonding by the pH, and that's simply because of those hydrogen ions. If the pH is off, a protein will unfold, and the quaternary, tertiary, and secondary structure are affected. Does not affect the primary structure. You can see here the ribbon is still here, okay? But the other levels are all affected by the pH. And when it's unfolded like this, we say that the protein is denatured. Now, in addition to pH, salt concentration can affect this. Temperature, just think about frying an egg, right? That white goes from a clear to the egg white. That's why we call it white when we eat it. But that's because the proteins have been denatured. Salt, you can think of that. We used to salt our food when we didn't have refrigeration. We used to have salted fish, and you use sugar for this too. But what's happening here is slightly different functions, but what's happening is that the proteins are denatured from the bacteria, so it doesn't go bad because it kills the bacteria. Also, the presence of metals, right? So metals, if our metal concentrations are off in our fluids, it will also affect the folding of proteins. So this is one of the ways in which metals can be toxic to us. There are others, all right? But if you're talking about things like lead in the water and how that is affecting that, metals work as a toxin in several ways, but definitely when it comes to protein folding, that's another thing that it does. All right. Now, the last biological molecule, the nucleic acids, these ones are, again, polymers. They are polymers of nucleotides. All right. These nucleotides can vary a little bit, but all of them have 3 parts. There's a phosphate group, which is the same for all of them. A sugar group, okay, that's a pentose. All right. And there is a nitrogenous base. All right, and that nitrogenous base can vary a little bit as well. All right. Now, with this, okay, we'll talk about this later. The way these things are put together, I didn't really talk about this in class, but I talked about the 3' end and the 5' end. The 5' end is actually the phosphate, and here's joined to the 5 carbon, and the 3' is actually the 3rd position on this sugar. But that's not important for this class. Just know that there's a polarity to this, meaning there's a sidedness. There's a 5' end and a 3' end. It becomes kind of important when we talk about these guys interacting with each other, all right? Now, the bases can vary, all right? We have 3 that are called pyrimidines. There's cytosine, thymine, and uracil. The purines are adenine and guanine. And, for our purposes, if you can remember C, T, and U, and A and G, that probably carry you enough because you'll probably remember, all right? Now, we have 2 types. We have DNA and we have RNA. RNA, that's the one, contains the instructions for producing RNA and ultimately protein, because I think you've gone through this before. So genes code for proteins. The RNA is the go-between because the DNA is in the nucleus, and the RNA takes that coding information out to the cytoplasm where we make our proteins, because we don't make the proteins in the nucleus. All right, I know I'm generalizing there, but we don't. All right, now we need those proteins, as you know, because almost everything in the body requires proteins to function. All right, so let's look at the differences between DNA and RNA. All right, so we'll look at them here. All right, and I have this wonderful chart. Hopefully you can look that over. That's on page 19 of my notes. All right, so DNA, when it forms, all right, it forms a double helix. All right, and RNA, on the other hand, is a single strand, and it will fold on itself, but it is one molecule. It is a single-stranded molecule. Now you can see that some of these bases are interacting with each other. Now, in DNA, what we're dealing with here, all right, so in DNA, we use cytosine, thymine, adenine, and guanine, whereas in RNA, we use cytosine, uracil, adenine, and guanine. All right, it's summarized in my chart. All right, and there's different numbers of hydrogen bonding that interacts here. So these interact via hydrogen bonds. All right, so when you look here, there's hydrogen bonds between these as well as here. Now, C will always pair with G, whether we're in DNA or in RNA. And in DNA, we have an AT match, whereas in RNA, we don't have T, but we have U. So it's an AU match, okay? And there's 2 hydrogen bonds between AU or AT, and there's 3 hydrogen bonds between GC. Now, the other thing that's going to vary is the sugar, right? So let's go back here. And our deoxyribose is what's used as the sugar in DNA, and in RNA, it's ribose, okay? So DNA is deoxyribonucleic acid, RNA is ribonucleic acid, okay? Now, I did this little thing, and hopefully you give this a go. I have a question on the study guide for the midterm, and in that, I have a little question for you to do the pairing, all right? So I want you to be able to be flexible enough to know that A is going to pair with T and G with C when you're talking about DNA. And with RNA, A pairs with U and G pairs with C. And I did this little exercise in class, if you were there, talking about this anti-parallel nature of DNA. So give that a go, all right? So the 3' and the 5' end always match, okay? And the pairing goes as such. And it's, if you take genetics, you'll learn more about why that's so important. I just don't have time in this class to do that with you. But I'd like you to remember this anti-parallel nature and the bonding pattern between these different bases. So have a look at the study guide and try that little question. And you can email me your answer to make sure that you're right, all right? Now, again, and we're going to go over this in when we do this in lecture, but remember our DNA codes for those proteins. We have to get that coding out into the cytoplasm where we make those proteins, and RNA is the go-between, and of that we have 3 flavors: mRNA, messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. But we will go over that later. I'm not going to test you on that yet, all right? Now, for good review on macromolecules that's not too stressful, I recommend this link. It's really great. It's Mr. Anderson. If you've watched the other ones, Mr. Anderson again, okay? All right, and I think that is it. And I know it's a long video, but I want you to be comfortable with all this material. So hopefully that does it for you. All right, and I've got to give my daughter some dinner, so I am going to stop here. Hopefully that helps.